Okay, I was getting all amped up to complain to somebody about the volleyball team, but it sounds like maybe they're done. Hopefully, again, that won't be an issue. Hopefully you could all hear me. I know it's kind of hard to hear in the back. Making the best that we can. I posted this at the beginning of the virus material. So you won't find it right where we left off. And this is a crude cut and paste from the Career Center website. But I wanted to make you aware that there is a lot going on in the Career Center these first few weeks of October. It's that time to, kind of think about already what you want to do next summer. And in terms of jobs, if you are still thinking about what you might do after graduation, if you're getting close to graduation and you think maybe you'd like to work in a lab somewhere, there are companies out there that hire college graduates that have lab experience, and they are very eager to have you. So I just wanted to make you aware of this. It's a Zoom thing that the Career Center is hosting. It's next week. Go into the Career Center's website. You have to register for it, but sounds like a great thing. My link got cut off there, but I did post it in your notes, so just want to make you aware of that. And I also wanted to start out here with a practice question from last time. We did a lot last time. Transcription and translation are fairly complicated. Thinking ahead to the exam a week from today, you will see a lot of questions that will try to confuse you the difference between replication, transcription, and translation. So here's an example. This is a question that I pulled from last year's exam. Do you remember what the Shine-Dalgarno sequence is? So take a minute, read over that, think about how you would answer. You can read that. All right, so the first two answers here, guess, what's the Shine-Dalgarno sequence? The first two answers here have to do with the initiation of transcription. All right, Pribnow box, don't confuse Shine-Dalgarno, Pribnow box. Pribnow box is an important sequence that helps RNA polymerase bind. These two, D and E, have to do, again, termination of replication, termination of transcription. Shine-Dalgarno sequence is that sequence on the messenger RNA that lines up with the ribosome. So that's important in the initiation of translation. So the answer there, correct one, was C. So that was a term that, again, was in your book. We talked about it in lecture. They don't talk about it very much in your book. But again, I encourage you to be working on that chapter 7 homework. We're going to be finishing chapter 7 today. There's a lot of animations that, again, take the time to watch those and understand them. More times you see something, the more likely you are to remember it. All right, so I'm hoping to finish chapter 7 today and get started with the viruses. And then your exam a week from today will cover through the end of the viruses. So I'm actually going to trim out a little bit of today's lecture because I want to make sure that we get to all our virus material. All right, we talked about how, in the last few weeks, how an organism gets ATP through different kinds of like aerobic respiration, anaerobic respiration, and we haven't really mentioned about how expensive it is for a cell to make proteins. It's actually quite expensive, so it's going to be necessary for the cell to conserve energy as much as possible. So a cell only wants to make the proteins that it absolutely needs. Just to give you an idea, do you have any idea how much ATP it costs a cell to make a regular, normal protein. A typical protein is about 300 amino acids in length. Okay, any ballpark guesses how much ATP that costs a cell? About 1,200. Okay, so if you think about, well, cells only getting, a couple ATP from glycolysis, 30-some from aerobic respiration, that's a lot of ATP. That's a lot of sugar a cell has to burn through in order just to make one protein or one enzyme. Okay, so it makes sense for a cell to try to conserve energy whenever possible. So let's think about how a cell might regulate, what it really needs and what it doesn't. We've mentioned a couple kinds of regulation. It might be built into the DNA sequence, how often that DNA gets transcribed and translated. We looked at the consensus sequences. These are, remember, I'm going to show you the graphic that we talked about, variation at the promoter region. Okay, so I'll come back to this slide in a minute, but remember this slide that we showed last time. This is the sequence of promoter regions of various operons of E. coli. So remember, an operon, it's a group of genes under the control of a single promoter, or the name operon came from operator. And we're going to review operons in a minute, but so under the control of a single operator, a single promoter. And we talked about these regions, the -10 region and the -35 region, in that sequence of DNA of the promoter region that the RNA polymerase especially recognizes. This is our RNA polymerase. And if it binds really tightly to especially these regions, then the RNA polymerase is more likely to transcribe the gene. All right, so we had a special name, Pribnell box, for that -10 region. If those two regions there are close to some consensus sequence, then again, the RNA polymerase is more likely to bind. Okay, so to some extent, there's a little regulation written right into the DNA. We also talked about the use of different sigma factors. Okay, remember, sigma factor is something that binds to that RNA polymerase and helps the RNA polymerase adhere to that promoter region. And a cell can actually use different sigma factors that help it bind more or less tightly. Okay, so more tightly it binds, more likely it is to transcribe. But we're going to take a little bit closer look at now at operons. Okay, and this just means because prokaryotes are polycistronic, they can have many genes on one messenger RNA, that genes can be regulated together. And this, they think that we may have some kind of operon type setups in eukaryotes, maybe a few here and there, but it's more of the exception. This is mostly a prokaryotic thing, where again, genes regulated together. They're usually genes that encode for proteins that would all be kind of used in the same pathway. So it's very efficient and makes a lot of sense. So I know that you've talked about operons in other classes. It's the sort of thing that needs a little review generally. So let's look at the parts of an operon. Just remember what this line is. I remember the first time I learned about operons in Bio 101, I was like the most lost that I've probably ever been in any lecture before. This is the template strand of DNA. So remember, when DNA is transcribed, we're only using one strand or the other. The strand that's used is a template. The other strand is called the coding strand. So here's the template strand of DNA. There's a promoter region. Remember, this is where the RNA polymerase binds. The operator region is where a repressor binds. Okay, and then you have the structural genes here that encode for certain proteins and usually enzymes in some pathway. The what's that? It should be in the second chapter 7. Okay, good. Okay, and then if there could be a regulatory gene upstream here that encodes for the repressor. All right, so those are the parts of a prokaryotic operon. We're going to look at several operons in particular, and thinking about how we kind of organize different operons. Your book doesn't really organize it in this way. It's actually cut a lot of material that deals with operons, but operons can be considered under negative control or positive control. So we're going to give 2 examples of operons under negative control and one that's under positive control. So basically, if an operon's under negative control, that just means that there's some repressor protein that's involved. Okay, and remember, this is usually the repressors bind to the operator region. Okay, and an inducible operon usually is going to be on. Okay, and that, sorry, I said that, I think I said that wrong. Yeah, okay, forget that, sorry. I get that mixed up. Usually an inducible operon is off, and that means not transcribed. Okay, so there's going to be some inducer that deactivates a repressor, and most often catabolic operons are regulated this way. So remember, by catabolic, I mean it's enzymes that are important to break some nutrient down, some bigger molecule. Okay, so the best example that you've heard of an inducible operon is going to be the Lac Operon. All right, so we're going to review the parts of that in just a minute. A repressible operon then is usually on, meaning it's usually going to be transcribed. That's its normal state. It involves some kind of co-repressor that would activate the repressor. And most often this is going to be anabolic pathways. So enzymes that are necessary in order to build some cellular component up. So the example that your book gives, let me spell that, is the trip operon. Okay, enzymes that are necessary in order for a bacterial cell to synthesize the amino acid tryptophan. Something that we as humans can't do, but a lot of bacteria can. All right, so let's look at the Lac Operon. I've tried to encourage those of you that have come to my office and ask, how do I study? It's always a great idea to be testing yourself as you study. Maybe you read over something and then you think, okay, now what did I just read? Could I explain that to somebody else? Okay, so I found a great example of the Lac Operon. I think this was just a little graphic off of Wikipedia where I could take off the labeling of it. I want you to take a minute or two and see if you can label the parts of the operon. All right, I know that it's probably been a while, but we read a little bit of it already. So however you want to do it, whether you want to number it off to the side or just write it in, just take a minute and see if you can figure out what all those parts are, okay? And then we'll come back together. All right, let's see how you're doing. So number one, the big yellow blob there, that's the RNA polymerase. Okay, and the spot where the RNA polymerase binds, yes, remember this is transcription, that's trying to initiate transcription. So number 3 there, that is the promoter region. Number 2, that denotes some kind of repressor. And we mentioned the repressors are always bound to the operators, the operator region. Okay, and when you look at this, okay, this really hopefully makes a little bit more sense here. The repressor here, when it's bound to the operator, it's a physical block. The RNA polymerase can't get to the rest of can't get to these genes here to transcribe them. Okay, so the repressor is keeping transcription from taking place. Number 5 down here denotes lactose or allolactose, whatever you want to call it. Allolactose just says slightly different linkages between the glucose and the galactose that make up lactose. So when lactose is present here, it's binding to the repressor, and so that now the RNA polymerase can transcribe the structural genes. Let's see, 6 is the structural gene beta galactosidase. I think it's LAC-Z. I'll just put that here. Oops. Let's see, 7 denotes LAC-Y. That's lactose permease. So beta galactosidase is the enzyme that cleaves the bonds between the 2 monosaccharides that make up lactose. Okay, that's absolutely necessary to have that enzyme in order for the cell to metabolize lactose and use it as a nutrition source. The lac permease, its job is to make it more easy for the cell to transport lactose into the cell. So the question you might think of, well, how can it make that enzyme? If only lactose is present, that's the only time it makes the enzyme. So how can it be present if it can't get into the cell? Well, the answer is it can get into the cell. It's just not easily done. A great analogy is if you've ever driven around Chicago, you know you want to have a fast pass, because with a fast pass, you can just drive right through the tolls. You don't even need to stop, and it's cheaper. Okay, so lac permease is like a fast pass for lactose. It can get into the cell really easily. If you don't have the lac permease, it takes longer, it's more cumbersome for the cell to transport in lactose, and it costs a little bit more energy. So just like when you're driving, it costs more and you have to stop at the toll booth. The 3rd structural gene here, LAC-A, that encodes for thiogalactoside transacetylase. And the mystery is, we know that the function of that protein has some kind of detoxification function, but I don't really know why that it has to be transcribed with beta galactosidase and lac permease. Like why it's really important in conjunction with lactose is kind of a mystery. So don't worry about the function of that. Okay, so hopefully if you remember, why the cell would want to make those enzymes, and hopefully it makes sense then it's only going to make those enzymes when lactose is present. There's no lactose in the cell, there's no use making an enzyme that cleaves lactose into its monosaccharides. Okay, the trip operon. And I've got your graphic from the book here for the trip operon. There's also a nice few minutes YouTube tutorial on the trip operon. So if you want another explanation of that, there's a couple of quiz questions at the end of that that are really nice. This is like I said, an example of an anabolic enzymes that are necessary for an anabolic pathway. So with this Operon, there's actually 5 structural genes making enzymes that are necessary for the synthesis of tryptophan. Okay, so like I said, this is normally on. So, you know, depending on where the bacteria is, assuming that it's somewhere in some medium where there's no tryptophan, it's an essential amino acid for the cell to make protein, so the cell always needs tryptophan. If, however, the cell is in some medium where there's already tryptophan, it doesn't make sense for it to keep making it. All right, so if there's lots of tryptophan present already, that tryptophan will bind with the an inactive repressor and then activate it to bind to the operator region. Okay, so if there's lots of tryptophan there, then it's going to halt the synthesis of the enzymes necessary in order to make it. Okay, so both the Lac Operon and the Trip Operon are examples of negative control because it involves a repressor. And whether it's normally stopping transcription or normally inactive, it's both considered negative control. Okay, well, I want to return back to the Lac Operon for a minute because the Lac Operon is actually a little bit more complicated than that. Because a cell, we talked about how cells only want to use, they always like to use glucose as their first choice of energy for metabolism. So if in the cell's medium there's both glucose and lactose, a cell wants to use glucose first, and it wants to repress using lactose. So even though lactose is there, we don't want to make those enzymes to break down lactose because the cell's just going to use the glucose. All right, so this involves another layer of control, positive control. There actually also needs to be an activator present for the lactose enzymes to be transcribed. And that involves this cap cyclic AMP complex. And this is not that complicated if you remember a couple of key things. So like I said, glucose must be absent for the cell to utilize lactose. Okay, and again, this cyclic AMP cap complex is an activator for the Lac Operon. And the next graphic shows that a little bit better, so hold on to that thought. Okay, the levels of cyclic AMP are related to the levels of glucose in the cell. Okay, so when glucose levels are high, then cyclic AMP levels are low. Okay, the amounts are inversely proportional. All right, so lots of glucose, low cyclic AMP. If there's low cyclic AMP, it doesn't bind to this cap protein and you don't get an activator. Okay, however, when glucose levels are low, then cyclic AMP levels are high. All right, so again, low glucose, high cyclic AMP, we get this activator, and then that's a signal that the RNA polymerase can bind. All right, so this graphic is helpful in the book, I think. Again, if there's no glucose, the cyclic AMP binds to this cap binding site. And this would be a little bit more helpful to understand that the RNA polymerase recognizes that, and it has to recognize that in order to transcribe those Lac genes. Okay. If you want a whole review of that lactose operon, including the negative and positive control, 5-minute YouTube video there, that's very good. And I didn't look at that again this week, but hopefully that's still up. My remembrance was that it was kind of spot on. So if you feel like you need a review of that, then look that up. Okay, so I just wanted to mention one other thing because it kind of reminds us back thinking about growth curves. We call this catabolite repression. So like I said, when a cell has both glucose and lactose available, it's going to repress using one catabolite, lactose, in favor of using another, okay, due to the regulation of the Lac Operon. So what we see, if we say inoculate E. coli into a medium with glucose and lactose, and you'll be doing this later on in lab using some mediums like that, not making a growth curve, unfortunately, because that takes a while. But if you then check the optical density, that's one way to check whether cells are growing or not using a spectrophotometer. See if the turbidity or the cloudiness of the culture is increasing over time. So you add your cells here at time 0. Remember, usually with any population just starting to divide, you get a lag period to begin with. So that's when the cell's kind of trying to decide what enzymes it needs in order to use whatever nutrients it has. And then it goes into a log period of maximal growth. When the glucose, though, is used up, we're going to get a second lag phase because now the cell has to kind of ramp up using a different nutrient, lactose. Okay, and then we get a second log period when the lactose is utilized. When that gets used up, then it goes into a stationary phase. Okay, so we call this, again, catabolite repression. The curve we can refer to as dioxic growth. Okay, basically two-phase, dioxic, two-phased growth, where we get, again, 2 lags periods and 2 log periods. Okay, so that's kind of cool. I have taught labs where we've tried to replicate this, and it just, E. coli replicates every 20 or 30 minutes under ideal conditions, so it really takes a number of hours, 5 or 6 hours, to really get to see this really well. Okay, any questions? Don't be afraid to jump out and ask if you do have a question. I want to talk just very briefly about mutation. So what happens when something goes wrong with replication or transcription or translation, and you get, you know, we get a different protein than what was intended by the original DNA sequence. If the DNA or RNA sequence permanently changes, we call that a mutation. Usually this is bad for the cell, right? Almost all mutations are going to impact the cell in a negative way. There may be, you know, a very small percentage of the time that it actually gives the cell some kind of advantage. We're going to talk about a little terminology to do with point mutations. That's when there's just one single nucleotide base that either is altered because the DNA polymerase III or RNA polymerase made a mistake, or maybe there was some chemical mutagen or maybe some radiation that caused that. Just be aware that there are other things that cause larger mutations that would involve, you know, multiple base pairs. We're not really going to get into that so much, but that can happen as well. All right, so I think this is the slide that I posted. It's kind of small to see here in class, so I'm gonna just break this up so that you can see it in class a little bit easier. But if, say, this is the normal DNA strand up here, the one that's used as a template for transcription of the normal RNA, and then this is the polypeptide, the sequence of amino acids that would result from that. And again, so each codon, if you look back at the genetic code, then you would see, oh, that UUU codon of messenger RNA corresponds to phenylalanine. Okay, so I don't have that memorized, but you could look back at the genetic codes to figure that out. Okay, what if there was a mutation right here, and you know that RNA polymerase doesn't make mistakes very often? We said like 1 in 10 billion, but if it did, and this was a guanine instead that happened during replication, then we would get UUC codon instead of UUU. But if you look at the genetic code, because there's wobble in that third codon, or sorry, that third base of the codon, that actually still corresponds to the same amino acid. So there's not going to be any change in the amino acid sequence due to that mutation. So we call that a silent mutation, because there's really no repercussions from that. Even though there was a mutation, it didn't cause any change. What if there was a mutation here? This G was changed to a C. Then we would get a codon GA instead of GCA. That would encode for a different amino acid, glycine instead of alanine. Okay, so we call that a missense mutation. It made sense in that it encoded for an amino acid, but it's not the right amino acid. So we call that a missense. So a slightly different amino acid sequence. It's hard to tell how much impact that would have. It depends on the protein. It depends where the amino acid is in the protein. It might not make any difference at all. It may have a huge difference. It depends totally on the protein, where it's at. However, if you had, say, this T changing from an A, and so then you now have the codon UAA, that actually is a stop codon. And so the amino acid sequence will be truncated after that one little point mutation. So that is typically going to be pretty bad, depending on if it was at the very end of the protein, maybe it would be fine. If it was at the beginning though, obviously the rest of the protein wouldn't get made. So we call this a nonsense mutation because it's almost for sure not going to be a functional protein that's going to result from that. All right. And of course, if you got some kind of insertion of a base pair or a deletion, that usually is pretty bad because it's going to knock off the whole reading frame. We call that a frame shift mutation. And everything after that insertion is going to be different amino acids than what it was originally supposed to be. So that generally is a typically pretty bad mutation as well, some kind of insertion or deletion. Okay, so just a little terminology there to kind of think about what happens. I think a few slides here that we're going to skip for now. So what causes mutation? Just for now, it can be caused by radiation. We're going to talk a little bit more about radiation in our next session when we're thinking about if we want to kill bacteria, how can we do it? Radiation causes lots of bad mutations in the DNA. That's why it's bad. But we're going to skip over this for now, as well as the chemical mutagens. We're not going to really talk about those at all later in the class. So I'm just going to skip over that for now. And if you look at the learning objectives, I posted a new learning objectives that just has a few crossed out. Okay, so mostly I want to skip over the repair mechanisms as well. In general, we have enzymes, bacteria have enzymes that go and repair any kind of missense, any base that was not replicated or transcribed correctly. Those are really important. Different kinds of enzymes. The one that I want you to not worry about these. The one that I want to talk about here, though, because we mentioned it before, is the mismatch repair. And mismatch error, again, is just when there's the wrong base inserted in there. So this depends on the level of methylation. We talked about that before. After DNA is replicated, eventually in bacteria, the adenine bases get methylated with a CH3 group. So here's a nice graphic, hopefully, that explains that. So here we have the old strands of DNA. You can see the adenine have ME. They could have put CH3 on it, whatever. The adenines are methylated. Here's the new strands. So remember, semi-conservative, we've got an old strand and a new strand that's making up the new DNA. Okay, and just to review directionality here, this top strand is going in this direction. It's always adding on to the 3' end. So this is actually the, what do you call it? The strand with the Okazaki fragments, the lagging strand, and this is the leading strand here, adding on to the 3' end. Okay, so say in your new strand of DNA, for whatever reason, the RNA polymerase sticks a thymine there adjacent to a guanine. Okay, that shouldn't look right. Remember, guanines always should pair with cytosines, and adenines should always pair with thymines. Okay, so we've got a mismatch there. So enzymes are going to come down the new DNA transcript looking for errors. And when it finds this error, how does it know which one to fix? Well, it's going to fix the new strand. It can tell that the G is the old strand because, again, it's going to detect the higher level of methylation on the old strand. And so you can see here that that T gets changed to a C. And then eventually both strands become methylated, hopefully after the repair enzymes have time to proofread everything. Okay. All right, so the last part of chapter 7 to think about, there's one other way that we've found that the DNA of an organism can be altered. And that's by horizontal gene transfer. So vertical gene transfer is when you know, a mother passes on its genome to its offspring. So in bacteria, that's, you know, when one cell becomes 2 cells, right, that the 2 daughter cells have the same genome as the mother cell, right? The horizontal gene transfer is when genes are shared with cells of the same generation. All right, and there's 3 kinds that we'll look at individually here. Transformation, and that's when recipient cells can take up DNA from the environment. So when would DNA be in the environment? Well, when a cell dies, its cell walls, cell membranes start getting leaky, and the cellular contents basically leak out of the cell. So there could be some DNA floating around from dead cells. Okay, we can actually make this happen to cells in the lab, or there are certain kinds of bacteria where we think this happens, you know, more often. So we'll look at a really important experiment that helped us understand that transformation can take place. Transduction, that's going to involve viruses that infect bacterial cells. And the third kind that we referred to before is conjugation, because that's mediated by a pilus. All right, so let's look at this really interesting experiment done by Frederick Griffith in 1928. So that was, in 1928, we knew there was something in cells that carried, you know, information. We didn't know that much more about it. Of course, we didn't understand the structure of DNA until the 1950s. So it's kind of crazy that he even did this experiment and that he understood what it meant, really, is quite impressive. But he took several different strains of Streptococcus pneumonia. This is an organism that causes a very severe pneumonia. You know, we get that as humans. It's very severe. Apparently mice are susceptible to it as well, and it usually kills them. So he had 2 different strains. The strain S was the wild type strain, and that's a strain of bacteria that makes really beefy capsules. So remember, a capsule can help an organism be more successful in infecting a host because the host's non-specific immunity, like white blood cells, have a hard time phagocytizing that bacteria if it has a big capsule around it. All right, so those cells were pretty successful. When he inoculated mice with that wild type strain of strep pneumoniae, it usually killed the mouse. But he had this other strain that was a mutant strain that he called strain R. For some reason, those organisms didn't make capsules. So he called them rough colonies, or strain R, because on a plate, those colonies would look kind of dry and wrinkly rather than shiny and smooth if the organism was a big capsule producer. All right, and when he injected organisms with that strain R, they didn't cause the mouse any harm. The mouse's immune system probably took care of those bacteria right away, and it didn't even get sick. He took some heat-treated cells of the wild type strain S, so dead cells, inoculated them into a mouse, and as you would expect, that didn't have any effect on the mouse either. So then what he did, he mixed some of those wild type, or sorry, not wild type, the living strain that didn't produce capsules together with the heat-treated wild type cells. So both of them separately don't cause any problems in the mouse. But oddly enough, when he mixed them together and injected them into the mouse, mouse died. So when he isolated some cells from the tissues of the dead mouse, he found living cells of strain S. Okay, so this was the evidence that cells, these living cells, strain R, took up some DNA from the dead cells, and this strain R was able to transform into the wild type capsule producing organism that again caused that pneumonia. And he found that he could make this happen in the test tube as well. He didn't need a living organism for it to happen. All right, so that's the evidence that we know that transformation takes place. We don't think that takes place very often. Again, it's probably certain bacteria that are able to do this. And we don't really understand how that DNA is transported in the cell. Right, DNA is really long. It just seems like it would be very difficult for a cell to transport that in, and that's not very well understood. Okay, transduction. Like I said, we're going to talk more about viruses very soon, but for now, just understand that this involves a bacteriophage. A bacteriophage is a virus that infects bacteria cells. And we're going to look at the replication of bacteriophages next time. But basically, when those insert their nucleic acid into a bacterial cell, they often will disrupt the chromosome of the bacterial cell because the virus wants to take over and make its own proteins. You don't want to make the proteins of the bacterial cell. And when the virus kind of assembles within the cell, it may accidentally get some of that bacterial cell's DNA in there and transfer that to another bacterial cell. Some viruses don't kill the cell right away. Sometimes they integrate some of their DNA into their host cell instead. So this virus might have actually brought some of DNA from one cell into another cell. And there's different kinds of transductions, specialized and generalized. But for now, just kind of know the basic idea that that is going to involve a virus. Okay, and lastly, we talked about conjugation a little bit already. Okay, what's special about conjugation is that this is the only kind of horizontal transfer where both cells are going to be alive. Okay. And this involves, like I said, involves a pelus, a conjugation pelus, right here. A cell that is able to make a conjugation pelus is said to be F positive. So remember when we first started in chapter 7 talking about genetics, we talked about how bacteria can possess lots of different kinds of plasmids. Okay, one of those plasmids that we mentioned was an F plasmid, a fertility plasmid, and that enabled the cell to be able to make a pilus. So if an F positive cell would be able to know somehow whether an adjacent cell, and it doesn't have to be a cell of the same species, it can be just kind of a similar species. If it doesn't have that F plasmid, it can reach out a conjugation pilus, draw those two cells closer together, and then exchange that plasmid. Okay, the F plus cell will share its plasmid with the F negative cell, and then the F negative cell becomes F positive. Okay, so the donor cell is F positive, and the recipient cell is F negative. Okay, so what's going on inside the cell? This graphic in your book kind of explains it a little bit better. We used to think the DNA was transferred right through that pilus, but we now think that the pilus just brings the cells close together and a pore opens up between them. Remember, DNA is always double-stranded. The plasmid of that DNA, it actually shares one strand of its DNA with the other cell, and then both the DNA strands in the donor and the recipient replicate to become double-stranded again. And now the new cell is F positive. Yeah, question? Go back to this slide for a second. There it goes. Okay, so kind of the what if here is not just that the new cell can now make a pilus, but the cell can transfer other plasmids as well. So if the cell has, say, an antibiotic resistant plasmid, now that cell can share that with other cells around it as well. So that's how certain traits can spread a little bit more easily. Again, we don't think this happens very often, maybe like to 10% of the cells, but bacterial conjugation is often sometimes termed bacterial sex. I don't think it's a great analogy because only one cell is sharing with the other, and of course there's no offspring, but it does, you know, intimate contact between two living cells, I guess. Okay, questions as we move on? Okay. All right. So again, starting today and hopefully finishing up on Thursday, I think the virus is certainly very applicable to our lives right now, thinking about viruses. Probably you're sick of thinking about viruses. So we're going to look first a little kind of introduction, characteristics of lots of viruses, how do we classify them. Probably next time we'll get into replication of viruses. Like I alluded to when I was talking about transduction, there's two different, a lytic cycle of replication and what we call lysogenic cycle, when the virus actually integrates its nucleic acid into the host. There are some differences in viruses that infect animals or humans with viruses that simply infect bacteria. And then we'll finish with a hopefully interesting discussion about prions, which are not viruses, they are proteinaceous infectious particles. So that's kind of a cool thing. So not for you to memorize, I don't think I posted this probably. This is just kind of a whole bunch of viral infections. And it would be fun to sit down and talk about, oh, I had this one and you had that one. Of course, viruses cause a lot of different problems. Some viruses, you might be infected with a virus and not even know it. Some of them will kill you 100% of the time, like rabies. You want to get rabies, that's a bad one. Interestingly, they put coronavirus here under gastroenteritis, which that just means you're throwing up or you're having diarrhea. That's gastroenteritis. So that's kind of surprising because even though this is, obviously an old picture, typically coronaviruses were more likely to cause a common cold. The Sars coronavirus here causes pretty nasty pneumonia. So I'm kind of surprised that they stuck it down there. But gastroenteritis, we usually refer to that as stomach flu. I always cringe though when somebody says they've been throwing up and they said, well, I had the flu. Like, well, that doesn't sound like influenza. Influenza is respiratory. Okay, so just remind me, hey, this is a time where you all should be hopefully getting your flu shot. I had mine last week. They are especially pushing flu shots this year because they're worried that if somebody has coronavirus and influenza at the same time, that could be a really bad combination. Now that we're moving inside and indoors more often, it's more likely for influenza to spread. Of course, when we have masks on, you would think that wouldn't happen, but they're just really especially worried about that this year. You do not want to get influenza. You are miserable for a week, body aches, high fever. It's respiratory, you're coughing. Okay, so when somebody is throwing up, that's not influenza, that's gastroenteritis. Of course, that's no fun either, but okay. So we have a hard time in general treating viral infections. We don't have as many drugs that are good. I will say maybe one exception there is HIV. We have good drugs. People that have HIV now are actually living fairly normally. So those drugs have nasty side effects, but we have developed good drugs for that. But a lot of these other infections, there's really nothing we can do. You just have to kind of ride it out for the most part. And I don't think I posted, I may have posted this, I forget. But this was the slide that I threw in there in February of this year when when I was teaching this class in February. And it was just interesting to me. At that time, there was about 400 deaths in China. And I brought it up in class. Hey, this is interesting. What's going on in China? We hadn't even decided on a name for this virus. We were still calling it the novel coronavirus. And even then in February, nobody knew what was about to hit us, right? That our lives would be totally changed. And I talked a little bit about the flu at the same time, not to say, oh, it's nothing, it's like the flu, but to say, hey, everybody's all worried about coronavirus, but really the flu, this causes all kinds of problems and it mutates every year and we never know how bad it's going to be, saying that the flu is a big problem too. So I just thought that was kind of interesting. Of course, viruses come in lots of shapes and sizes. Any virus that I talk specifically about right now, don't worry about memorizing that. I mostly mentioned specific viruses to just sort of make the discussion a little bit more interesting. But we will talk about some specific infections a little bit later in the course. Right now, we're just kind of in general characteristics of virus. Of course, here's Ebola. That was big for a while. Still could be again. We don't have a great vaccine for that. I don't even think there's a vaccine been approved for that yet. This actually is coronavirus. It's been in my notes for many years. The bacteriophage, the ones that infect bacteria, some of them have this really strange structure. It almost looks like a lunar lander. So we get lots of different shapes of viruses. And thinking about the size of them, of course, we haven't been able to visualize viruses until about the 1930s when the electron microscope came out. So before that, we knew that there was some small particle that was causing disease. We just couldn't visualize it. How small are they? Well, if you compare, here's a red blood cell here compared to an E. coli. Of course, usually bacteria are maybe 10 times smaller than eukaryotic cells, kind of on average. You can't even see the viruses very well right there. In this picture here, then, there is some variation, obviously, among viruses as how big they are. The smallpox is one of the bigger viruses. And then polio is one of the smaller ones. Some of the bacteriophage are pretty tiny as well. So maybe poliovirus is on average about as big as a bacterial ribosome. Just kind of get a sense of the size there. Okay, we won't be growing any viruses in lab. We used to do a lab with bacteriophage, but we had trouble getting it to work consistently, so we threw it out. Remember, you need living cells in order to grow viruses in. So you're not going to get viruses growing up on your plate of media in lab. And we'll talk a little bit later how we do grow up viruses when that's necessary. All right, so general characteristics of viruses. They always have nucleic acid. So that's going to be DNA or RNA, one or the other, not both. Okay, so really the more we learned about viruses, this was, here's a particle that doesn't have DNA, yet it can reproduce. So again, DNA or RNA, not both. That nucleic acid is going to be protected by a protein coat that we call the capsid. Okay, so this is a protein shell that protects either the DNA or the RNA. Okay, and it's composed of capsomeres. So all these little kind of round thing here, those are the capsomeres that make up the capsid, their protein. Okay, and it can be the means of attachment to the host. So there might be, certain proteins here on the surface of the capsid. They're going to be specific for certain host tissue so that it can attach and invade. Some viruses are going to be enveloped. Okay, they're going to have a phospholipid membrane surrounding the capsid. Okay. So we'll see a picture of an enveloped virus on the next page there. They typically are going to pull that membrane from one of the host membranes. So either the nuclear membrane, more often the plasma membrane. As it leaves the host, it's going to take that with it. And a little terminology here, when a virus is outside of a cell, it's more correctly called a virion. So the virion is going to be the nucleic acid and the capsid if it has an envelope. When it's inside a cell, it's more correct to call it a virus. So when it's inside the host cell, the envelope is going to be degraded or gone away. The capsid has fallen off I forget the word, I'm losing the word there. But it really just involves the nucleic acid. That's the virus inside the host cell. Okay, the envelope and the capsid are going to be removed. And I try to use those terms correctly, but it just is more natural to just say virus all the time. So sometimes I slip into that. Okay, so just like when we first started talking about characteristics of bacteria, we talked about morphology like cocci and bacilli. It's a little harder to do that with viruses. We can kind of loosely put them in 5 categories, either helical or enveloped helical. Here's an enveloped helical. So you can see the capsid is helical, but if you looked at the outside of the cell, it wouldn't look helical because there's a membrane around it. And by the way, this is the Sars Coronavirus. That's a picture from your book. Sars Coronavirus is about 9% mortality. So not as transmissible as what we're dealing with now, but more deadly. And luckily, that outbreak was in like 2003, and we haven't really seen much, I don't think, at all of that since then. But that's a little bit about severe acute respiratory syndrome. Polyhedral morphology, we're going to take a look at that, envelope polyhedral. And then there's another category that just doesn't really fit into any of those other categories. All right, so let's look at examples of each of those. If the capsid is helical, or this is viruses that, you know, basically look like little sticks, long and thin, like the tobacco mosaic virus. That was the first virus that we saw under the electron microscope. Influenza virus is an example of a helical capsid that's enveloped. Okay, so kind of looks more roundish. Okay. My slide kind of flopped in on top of itself here. Sorry about that. So a polyhedral morphology, that just means the capsid takes up a many-sided shape. Most common is the icosahedral symmetry of that capsid. And I actually don't know if there are other shapes other than icosahedral. I haven't come across an example of that, but maybe there is somewhere. An icosahedron is a 20-sided shape. Okay, so you have 20 faces here with equilateral triangles are each of the face. Okay, so that structure is most common because it gives the capsid good strength because it's so symmetric. Okay, very stable, very strong, again, because of these equilateral triangles on each face. So this is the most popular kind of polyhedral. It has, if you look at the capsomeres, it has penton proteins as well as hexon proteins. The penton proteins, I got a little, whoops, sorry, circled the wrong thing. Little diagram over here. The penton proteins are the ones, the capsomeres are at the corners. Okay, there's always going to be the same number of those. There's always going to be 12. So if you look at this, this is actually an adenovirus. Over here, you can see the blue ones here are at the corners or the vertices. There's always going to be 12 of those. Adenovirus, that causes a lot of different kinds of things in humans, like pink eye, colds, bronchitis. It was named because it was first isolated from human adenoids. Okay, so that's an example of a virus just that's naked. It doesn't have an envelope. The hexon proteins are the capsomeres on the faces. Okay, so the red ones over here talks about hexons right here. There's always the number of hexon proteins is going to depend on how big the virus is. If it's really big, it's going to have more. If it's smaller, it could have a smaller number. Okay. Let's see. All right. Enveloped polyhedral. Yep. My writing's a little bit sloppy there. Capsomeres. Let me try spelling that again. Capsomeres. Okay, on the faces. So here's the hexon capsomeres. I'll draw them in there. All right. And then you can have an enveloped polyhedral. An example here would be herpes virus. You've got, again, this polyhedral capsid. It's encircled with an envelope. Herpes is a little bit more complicated because it has these other proteins called tegument proteins. A few viruses have those, but it's pretty prominent in herpes virus. Okay, and then I mentioned there's a few shapes that really don't fit into any of those categories very well. So we just throw them into the complex category. Rabies here has a very distinct bullet shape that's visible right away under the electron microscope. Here's smallpox that, again, just doesn't fit any of the other categories very well. Bacteriophage here, again, weird lunar lander shape, even though it's got a icosahedral capsid. It's got these other tail fibers that's just most of the time they're considered complex. All right, let's look a little bit more closely at that viral envelope. Okay, which viruses have envelopes? This is going to be much more common in viruses that infect animals. Okay, so humans is what we're most interested in, so I guess I could replace that with humans. Because typically the way they replicate, it's just more common, they grab it from humans. Bacteriophage more often burst a cell, so it's harder for them to take bits of the bacterial cell's plasma membrane, so that might be one way that you could think about that. Because they acquire them from the host. I talked about this already. The membranes are the same composition as our membranes. And oftentimes there's going to be some kind of glycoprotein spike. And this can be, like here, our adenovirus has some spikes. I didn't mention those before. This can be attached, right from the capsid, or like an influenza virus, they can be stuck in the envelope. And those spikes, sometimes these are called pepilomeres as well. I usually just refer to them as spikes though, but you might see that term somewhere else. Those spikes are very important in recognizing host cells, attaching to those host cells, and gaining entry into the host cell. So for example, influenza have these protein spikes, hemagglutinin and neuraminidase. We're going to talk about that in more detail later. It's just sort of an example now, but you might have heard, flu strains, H1N1, for example. That's named for the different variations of these proteins, hemagglutinin and neuraminidase, on the surface of that influenza virus. There's different variations of those. And those spikes are usually what our vaccines, we take a little bit of that protein from the spikes to try to stimulate our immune system, because that's the most antigenic part of the virus, or the part that best stimulates our immune system. Okay, so if a virus doesn't have spikes, again, there would just be maybe proteins embedded in the membrane or in the capsid that would recognize specific hosts. Okay. Oh, and one other thing that I wanted to mention here, thinking about how hardy viruses are when they're just in the environment. So meaning they're not in a cell. If they're in the water or say on your desktop, how hardy are they? Sorry, I think it's harder and hard. Oh, whoops, sorry. What did I hit? Sorry about that. Let's see if I can get it back. Okay, somehow, I don't know, I must have bumped something. Oh, there it is. It was just slow. Typically, my writing is messed up. Okay, so what I was about to say is that viruses that have a membrane are typically really wimpy in the environment. And you might think that that's not intuitive. You would think maybe a membrane would protect them. But think about it, they're phospholipid membranes, they dry out really easily. They're really susceptible to you know, sunlight to totally drying them out. So a membrane virus like coronavirus or influenza virus on surfaces typically maybe last hours up to a couple of days. That's considered, you know, fairly wimpy. A virus that does not have a membrane, like norovirus. Norovirus is one that's nasty thrown up and diarrhea, usually at the same time. It's terrible. Norovirus, it does not have a membrane. That virus can stay viable for weeks on surfaces. Okay, so I guess the fact that coronavirus is membrane is a good thing in that it doesn't last forever, you know, on doorknobs and stuff like that. Even a day or two is longer than what we would hope it would. But that's, again, something to, kind of a general rule that we can go by. Okay. Okay, everything's all going to be goofed up here. I don't know what I did. So I think that's a good place to stop. We will hopefully finish our viruses next time. We'll get into some replication cycles. If you have any questions, don't be afraid to email me, and we'll see you next time.